Okay. Is that good? Can everyone see? Nobody's going to fall asleep? For those of you I haven't met, I'm Margaret Smith. I'm the maize or corn breeder in the department. And so Vern Grayson had asked me to speak today about breeding maize. I'll be trying to kind of target this to take advantage of some of the things you've heard about already. But if I start rambling on about methods you don't know anything about, feel free to stop me or interrupt me, and we'll explain them at the time. If we don't finish everything, that's okay. Also, I think you guys have a course website, right? So I'll send the PowerPoint to Luis, right? And so you don't have to feel like you have to write down everything that's in the slides, because you will ultimately get a copy of this PowerPoint. I love starting off with my collection of corn gems and candy corn. Breeding maize is fun. It's not quite as fun as getting gems out of it and candy out of it, but I still like it. What I'm going to try and cover today is first a little bit about the biology of the crop. Just, what is it that makes one make the choices we have to make in maize breeding? Some of the objectives, because those are always the thing that is going to guide your breeding choices. We'll talk a bit about breeding methods, since this is a methods course. And I'll cover inbreeding and heterotic groups, a little bit about testing, and a little bit about population improvement, because all of those are methods that one uses in the world of maize breeding. And finally, if we have time, lastly, I'll talk a bit about some of the work we're doing in my program to address niche environments or specialty traits. So that'll be the quick for fun at the end if we have time for it. So with that, let's launch right into maize biology. The first thing that one wants to know if you're starting off a breeding program is where did the crop come from? What is its pollination biology? In the case of maize, its wild relative is teosinte. And this picture shows a farmer's maize field in Mexico, actually. This is the maize. And right here, you can see a teosinte plant. They're all intermixed in farmers' fields. So maize originated from Mesoamerica, this area of Mexico and Central America. And as far as we know, it's still controversial in some circles, but as far as we know, its closest wild relative and its predecessor is teosinte. This just shows you the modern day distribution of several of the different teosinte species. The one that appears at the moment to be most closely related in terms of ancestry to maize is this kind of blue-green one in the middle here, Parviglumis. But they all are cross-compatible. They're all Zea species. All of them are cross-compatible to varying degrees with maize. So here's a whole set of wild relatives. It's a potential source of genetic variability. So one of the things for any crop you always want to know is where are your sources of genetic variability. The obvious ones are other varieties or other breeding programs with that same crop. And then the next step removed would be the tertiary or secondary gene sources, the exotics and the wild relatives. So you find that Mexico and Central America is the area of origin. It's in some ways quite amazing that maize was developed at all. The teosinte, which you can see on the right, this is actually the seed case from a teosinte. So you can see it was quite small heads. Each of these contained maybe 10 very small seeds, about this big, if anybody can see that space among my fingers. And they were completely indigestible. So cast your mind back to the original plant breeders, who were the first people that began domesticating crops, okay? For people who began trying to plant instead of just gathering things. They actually carried out the first processes of plant improvement. And here you have this thing that has completely indigestible seeds that fall apart on the ground, and they're really hard to see because they're little black things, right? So who would have thought to try and domesticate this? It's really quite amazing. I mean, it's a fun exercise to do with many of our crops and say, okay, if I were a domesticator now, is this what I would pick? Well, maybe not, but on the other hand, this is basically what those early domesticators managed to achieve. That's a significant plant breeding improvement, right? The ears weren't quite this big 7,000 years ago, which is our first earliest recorded evidence of maize as we would know it today. They were smaller, but still a major improvement process. The early domesticators selected for several things that are almost always selected as part of the domestication process. Larger size of the edible plant part, in this case the seeds. Easier harvesting, so reduced shattering. More digestibility, things that were easier to process. So in this case, the teosinte lost its indigestible seed coat. And ultimately, you ended up with a crop that has a huge cob, hundreds of seeds on each cob. Probably this one has about 750, I'd say, as opposed to 10 on that. Enormous number of seeds, completely digestible, nicely enclosed in a husk until you choose to harvest it, so that helps protect it from pests, right? And they don't fall off. They stay there until you want them, until you shell them off and eat them. So that's a major improvement process. It also has created a crop like many of our crops, but perhaps more so, that cannot survive in nature. I mean, think about what has been lost. I just told you what had been gained in terms of things that people selected for in that domestication process. In terms of what has been lost, what traits that would be adaptive in nature have been lost from this crop? There's one biggie. Yeah? The ability to seed itself, a dispersal mechanism. So picture what happens if you just had this thing growing out in a wild field. You occasionally see corn in just field situations, all right? It grows, it produces its ear, it matures, the ear falls on the ground, right? Either that or a deer, in Ithaca, forget that. A deer eats it, okay? That's the end of it. It's completely digestible, it's gone. Or a bird eats it, or a raccoon, or a squirrel, okay? It's all gone. If not, if it manages to survive all that, it falls on the ground and maybe sits there until spring if nothing else eats it. And then what happens in spring? Maybe it molded because it has no hard seed coat to protect it. So maybe it molded, it's all dead again. Or maybe it survived all that and it actually germinates and you have 750 plants germinate in a space this large. And what happens to them? They all outcompete each other and they die. End of the story. So extremely poorly adapted in nature. The other way you can view this, and if you want to read an interesting account of that, I would commend to you Michael Pollan's book, The Omnivore's Dilemma. Has a really nice chapter about corn that'll make you think about a lot of things. But one of them he talks about that I've always liked to talk about as well is who domesticated who here. The other way to view this is that maize evolved a very nice symbiotic relationship with humans. Took very good advantage of us in a natural selection sort of way. Because what do? We harvest these seeds, we shell them off and put them somewhere safe from molds, pests, etc. We keep them at a perfect temperature to maintain their viability. The next spring we make a field that's nicely cultivated, we eliminate all the weeds that might compete with it. We seed the seeds nicely spread out so they're perfectly distributed and they have all the resources they could wish to grow. And they go ahead and produce a bumper crop. So we are the dispersal agent that maize has adapted to, and we do a really nice job for the crop, dispersing it every year. So you can think about it as a crop that has evolved to take advantage of us as the dispersal agents rather than kind of random gravity and animal feeding. And you can also think about it the flip way. Maize would not survive in nature, so it wouldn't survive without us. How long would we survive without maize and our other domesticated crops? Anybody care to raise your hand to think you could survive in nature right now without crops and livestock? So who's been domesticated? Food for thought. I'll stop rambling on about that. At any rate, maize, the teosinte is the wild relative of maize. It is fully cross-compatible, so therefore it is an accessible gene pool that you can use for traits for genetic improvement. Yeah?
It goes back and forth between saying it is and most people think it is. What's the contention?
There's always a little controversy about the origins of maize. There are other relatively closely related species. Tripsicum is the primary one. So there are some people who believe that Tripsicum also had a role to play in this. There are other people who believe that teosinte was not the ancestor at all, but that these were two distinct evolutionary branches from a ancestral species that no one has ever found. They hold that view very strongly for some reason, even though we have yet to find any evidence of that. So for some reason, I'm not entirely sure why, the people who are interested in maize evolution are very evangelical about it. They get their idea of how it happened, and they will argue, classic scientific table-stomping arguments about the origins of maize. Some of them are the rhetoric is very impressive. Most of the evidence, and particularly now that people have been studying more and more the molecular genetics underlying domestication genes in maize and finding some of those loci in parallel in teosinte, more and more the evidence is weighing, in my mind, on the side of teosinte really being the direct ancestor. But it's not without controversy still. Would you agree with that? I'm looking at you because I know you've delved into some of that literature too. Okay. So the biology of maize, it's monoecious. What does that mean? Anybody remember what monoecious plants are? Separate male and female flowers on the same plant, okay? Do we have a pointer? No. All right. Well, I'll just keep waving my arms. So the tassel up at the top is the male flower. The ear is the female flower. The way the ear works is the silks that you all know from sweet corn that come out at the end are the stigmas and styles. Each silk is connected to one ovary, which is the thing that will become the kernels, okay? So when the silks come out, there's one silk connected to each of these putative kernels. In order for maize to get fully fertilized, you have to have pollen grain land on every silk and grow down that silk to each of these individual kernels. So that was a lot of pollen grains that had to land on that ear. It's naturally cross-pollinated, as you might imagine. The pollen is shed up there at the top of the plant. It's a wind-blown pollen, but fairly heavy, so it doesn't travel for miles and miles and miles in any great amount, but it does tend to blow to something other than the ears silking right underneath it. Okay? There are very few fertility barriers in maize, so pretty much you can cross anything with anything. The one I have noted here, there are some gametophyte factors, and those are factors which, they're genetic factors called GA genes, and they will selectively exclude pollen. If a plant carries a dominant GA, it will selectively exclude pollen from plants that carry the recessive form of that allele. So that is one fertility barrier, and it's one that's derived. Most of the GA factors we now have in maize have been found in teosintase. So people discovered that if you took maize pollen and tried to pollinate teosinte with it, sometimes you would get essentially no seed set, whereas the exact reciprocal cross, the same teosinte plant onto maize silks, would give you good seed set. So obviously the two are cross-fertile, but there was something about the direction of the cross. And what they found was that there are gametophyte factors in some of those teosintes. So if you want to use them, you use the teosinte pollen onto the maize, and then you'll have in your population a gametophyte factor segregating out that you need to manage. These gametophyte factors, by the way, have also been very useful commercially. This is a nice case of a gene from a wild relative that has proven very useful. They're used largely for popcorn and white corn production in temperate areas. Why? Popcorn quality is largely a function of endosperm characters. What's the endosperm derived from? It's triploid, right? So one genome comes from the male, and two genomes come from the female, okay? So if quality of something like popcorn is dependent on the endosperm, it will then be influenced by what pollen pollinated that particular seed, because the pollen contributes one genome to the endosperm and therefore affects endosperm characteristics, hardness, and so on. So if you're a popcorn producer, usually it's a fairly small acreage because the world doesn't eat that much popcorn. And you're probably in the midst of a whole bunch of producers growing US #2 yellow dent to feed cows. So you have all this pollen coming in from dent corn that's going to alter the quality to the extent that there's any cross-pollination. It will reduce the quality of your popcorn. One way to minimize that is to build a gametophyte factor into the popcorn. And then even if it's surrounded by dent corn pollen, most of that pollen will not compete with the self-pollen from that popcorn variety. And this is exactly what's been done. So most popcorns grown in the US carry a gametophyte factor in order to minimize cross-pollination with standard dent corn. Okay? Same is true for white corn. Again, it's an endosperm character. If you're producing white corn, which is usually for food, so the color and the purity of the color is important, and you're producing it surrounded by fields of yellow dent corn for cow feed, you don't want all sorts of yellow kernels in your white corn. The gametophyte factor helps exclude that yellow pollen. So this is the only fertility barrier that we're really aware of. Those are ways it's been used commercially. In terms of breeding, it will influence which way you need to make a cross, but there are almost no crosses I know of that you cannot make in maize. Okay? And usually the only case where you need to worry about it is when you're making a cross with a teosinte. So maize is easily selfed and easily crossed. So it's a great organism to work with. You can do whatever kind of pollinations you want with very few problems. Also, maize biology is characterized by lots of diversity. As maize moved around the world, different cultures selected different types to meet their food needs and preferences and their different environmental needs. That diversity is certainly apparent visually as you look at a collection of lots of different maize varieties from around the world. You see all these differences in seed type, endosperm hardness, seed color, ear shape, etc., etc. Also differences in adaptation. Maize is one of the crops that's grown over the widest range of environments in the world. So you find it from the equator up to areas north of here, so well into Canada, as in commercial production. And you also find it from sea level and in fact even below sea level in like Lake Baikal, which is that lake basin is now dry, a lot of it dry, but below sea level, up to over 3,000 meters in altitude in the Andes. So it's grown over a tremendous range of environments, highland tropics, lowland human tropics, temperate areas. And all of that creates diversity because the crop has adapted to those various different environments. If we look at maize varieties with random markers, one of the things that we have seen so far, and I should emphasize that this has been with a fairly small number of varieties, not a huge number, is that within a variety, a heterogeneous open pollinated variety, okay, there's quite a lot of marker diversity. But for any one variety, that diversity is not so much smaller than the diversity of, you would find if you looked at like 30 varieties. One gets the sense, in terms of the genetic structure of maize germplasm, that what we have are a lot of open pollinated varieties that have kind of been a mixing pot historically. So lots of cross-pollination has occurred among them as they have moved and been selected. And that what they've been selected for are key traits, certain key traits that give them particular ear characteristics or particular adaptational characteristics, but that you don't necessarily pick up when you just look at them with random genetic markers. Those are probably fairly specific genes. In this case, that farmers saving varieties will select for particular ear types. So there's a stabilizing selection there that maintains those loci, those relatively few loci that govern kernel color, kernel texture, and so on, fixed. And the environment will always select for adaptation to that environment. Things don't produce seed if they're not well adapted. So that stabilizing selection also occurs. But then underlying that is all this mix of diversity. Some of it in expressed gene regions, a lot of it in unexpressed molecular areas of the genome, what people call junk DNA, but we call it that just until we figure out what it's really there for. So that's the kind of the nature of maize in terms of its biology. Tremendous amount of diversity to draw on within the cultivated species, wild relatives to draw on if you need. And of course now in the age of genetic engineering, you can draw on pretty much anything you want for genes as well. That's a lot more challenging. So let's move on to breeding objectives. I've sort of sorted this out. I have US breeding objectives, and then I'll talk more about breeding objectives in places like the tropics. This would not pertain only to the US, but I use the US because that's the focus of a part of my breeding effort as typical of temperate areas of the world. In the world of dent maize, dent corn as opposed to sweet corn or popcorn, basically the objectives are yield, 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 and yield. I had a breeder from the private sector tell me, okay, the 10 most important objectives in maize, breeding objectives, 9 of them are yield, and 10th on the list is standability. If it yields a lot but falls down, that's not so helpful. So really the game is about trying to improve yield. Why? This is a crop in the US and in most temperate areas that's largely for animal feed. Animals don't typically tell us a whole lot about preferences. So, you know, basically we're trying to give them an energy crop that produces a lot of energy per acre. So that's the goal in the US, temperate dent corn production for industrial or animal uses. Maturity is also important. Maturity because you need to have something which fits the growing environment, and obviously with a crop that's grown from, you know, Maine to Florida to California, you're going to have differences in growing season length and maturity that you need to fit. Plant health is always important. Pretty much any breeding program on any crop has some component focused on things like disease resistance. These, in the case of corn in temperate areas, are really about achieving that yield potential. If you don't have the right maturity, even if you have lots of genes for yield potential, you're not going to get that yield because the thing won't mature. If it dies of diseases or insects, you're not going to get that yield potential that you have because the pests took it away from you. We are now beginning to see an interest in some specialty types focused on things like nutritional quality, either for silage or for, in some cases, industrial uses or human food uses. And in the U.S., a recent phenomenon, last 5 years, we're suddenly seeing a dramatic upsurge in the use of maize for ethanol production. And that's just beginning to have some influence on the breeding process in terms of people thinking about whether there are ways we can focus not just on grain yield, but actually sort of the next step in this case, which is yield of ethanol from a given amount of grain. So you can measure yield in terms of grain per acre, or in this case, you could measure it in terms of ethanol per acre, which might be a combination of how much grain and how good is the grain quality for ethanol production. Okay, so that adds a layer. Okay, the tropics is sort of a different matter. And when I talk about the tropics, most of my experience in the tropics has been working not with large-scale commercial farms, but with smallholder farmers who are producing maize for food. What we would call dent corn and what we would think of as something only good for animal feed is actually the staple food for many people throughout Latin America, Africa, and even some parts of Asia. So a tremendously important staple food crop. If this is the crop right here that your family depends on for food, you're probably going to be interested in yield stability first and yield potential secondarily. Why? It's most important that you have, if you have 1 acre or 2 acres to produce the food for your family, no government crop insurance, no government assistance, nothing else to rely on. The first priority you have is to have food every year. So yield stability takes on a role more important than optimal yield under ideal conditions. Most years you don't have ideal conditions. There's going to be drought, there's going to be fertility stress, you're not going to be able to afford fertilizers, a whole range of things. So yield stability, despite whatever stresses that crop is going to face, is probably more important than breeding for optimum yield under ideal conditions. What plays into that? Of course, biotic and abiotic stresses. These things are often much more severe in the tropics for a whole range of reasons than they are in temperate areas. Warm temperatures, excessive leaching of soils that reduces fertility, lack of access, either just simply lack of availability or no money to buy things like pest control materials or fertilizers and so on. So biotic and abiotic stresses. And then a whole other realm that we pay very little attention to in this country, post-harvest and processing traits. So if you're storing your maize this way, you better worry a little bit about storage pests. You can have a great yield, but lose a lot of it to pests. If you're processing it by hand, your little girls are shelling the kernels, you better worry about how easy it is to shell, or they're going to get real tired real fast trying to do that job. So those post-harvest traits that as breeders we have tended not to focus on a lot, you know, the yield mentality, once I find out how much yield per acre I got, that's the end of the story. That is not the end of the story for many people. The end of the story is, okay, and does that yield last until next year's harvest in my storage? And is it producing the kinds of foods that I want in a reasonable way? So sort of a slight difference there in objectives. Before we move on to talking about methods, I do want to talk about the 2 main varietal types that we breed for in the world of maize. Open pollinated varieties, and you guys may have had this already. I'll cover it very quickly. If things aren't clear, please ask. An open pollinated variety is a genetically heterogeneous and very heterozygous variety. It has every plant in it probably being genetically distinct. It's maintained by harvesting ears out of a field of that variety, planting that ear out. The ear will be a product of, in this case, this female parent plant, and whatever pollen happened to pollinate those silks. So in the case of an open pollinated variety, what you have is a genetically diverse field. Each plant is a different hybrid. Okay? So each plant is the product of some kind of a cross between that female and whatever pollen grain happened to land on that silk. The difference from a hybrid is that each plant is a different hybrid in that field. Okay? That's the open pollinated variety. Hybrid varieties, there are several different types. We'll look at the simple case, the single cross hybrid first. In this case, what breeders are aiming to do is rather than have a mix in that, like that open pollinated variety where every plant in the field is a different hybrid, what you want to do is genetically create the constitution of one of the best hybrids in that field and have every plant be identical to that best hybrid. Why? Well, if you have a mix of genetic types, you're going to have sort of a distribution of yield potential with, you know, a bell curve with a mean in the middle. But if you can replicate over the entire field one of the best of those hybrids, your yield is all going to be over here on the high end of that bell curve. You're going to have higher yield potential, right? So in the case of hybrids, what you do is you inbreed, you get things which are genetically homozygous, and then you figure out which two of them crossed together will give you that outstanding yield potential. The seed sold to farmers then is a controlled cross between two different inbred parents. One of them somehow detasseled or emasculated genetically, one way or another. The other one serving as the pollen source. So that these ears harvested from the field and sold, that seed sold to farmers, you know is entirely a cross between this female and that male because there was no other pollen source in the field. There are more complex types of hybrids. I've just put a modified single cross up here, but some things where you make a cross to use as the female parent and then an inbred as the other male, or a double cross where you have hybrids on both sides. So there are lots of other types of hybrids, but that's the basic other varietal type is hybrid varieties. The difference between those two clear? OPs and hybrids? Okay, good. So in terms of breeding methods for hybrids, there's a fundamental challenge with a cross-pollinated crop like maize. So you have a cross-pollinated crop. Your need in breeding is to inbreed, something that typically happens easily with self-pollinated crops, but not with cross-pollinated crops. They don't do that job for you. Your most important objective, and here I'll focus on the US, is hybrid yield. And yield is generally low in heritability. How much have you guys talked about heritability so far? A little? A lot? Some? Medium? Okay. You have the general idea. Heritability has to do with the genetic, and particularly the additive genetic variance as opposed to the total variance that you see out there. So yield is generally low in heritability because it's highly influenced by environment. Okay, so there's a lot of environmental influences which reduces the genetic share of the variance, you know, because environmental variance is high. And also in maize, there are many dominance and epistatic effects, in other words, non-additive genetic effects that contribute to yield potential. So your additive genetic variance is a smaller share of the total genetic variance for yield. So all of that adds up to reduced heritability. Okay, why is that a challenge? Well, the fact that you have a naturally cross-pollinated crop, but you need to inbreed, it means you have to think about pollination control every single step of the way. Unlike, what crops have you guys heard about just recently? Let's see. Sorghum. Yeah, unlike sorghum or unlike rice, which Luis will talk about next time, you can't just, easy job there. He just plants out his plants and they self themselves for him. He doesn't have to do anything, right? So remember that when he's talking about how hard rice breeding is. You just plant them out, they self. Maize, you need to worry about pollination control for every step. So every self that is done, you must actually physically make it, okay? So what that means is you do your numbers a little differently. I'm not going to plant out an F2 population of 1,000 plants because it's ludicrous to think that I'm going to make that many selves by hand in one F2 population. In wheat or rice or other self-pollinated crops, you might easily do that because you can just let them do it themselves and then choose the plants you happen to like. So it makes a difference. Next, the fact that what you're working on as a breeder is inbreds, but the most important trait is a trait that's manifested on hybrids, means what you see as you're breeding is not what you're going to get in your product. Okay? So you're breeding along with something, but the final product is going to be something that's a cross with that, and for which looking at the inbred itself gives you relatively little idea of what the hybrid will do. So for hybrid yield, what you got is also hard to measure. Not only can you not see it on the inbreds, but yield is a very environmentally influenced trait. So it requires plots with significant numbers of plants, replications, multiple locations, a lot of sampling of the environment to measure that trait. So it's costly. So therein, you sort of have the challenge to breeding maize. You got to make every cross, whether it's a self or a cross, by hand. What you're looking at as you inbreed is not what you're aiming to get as a product variety when you're working on hybrids. And what you're aiming to get is a trait that's fairly hard to measure and takes a lot of resources. Okay, so what does that mean? In the world of maize, we use a lot of inbreeding methods. Okay, so the inbreeding methods that are most typically used are pedigree, and that you would use when you're making crosses of elite parents by elite parents. So you're basically taking the best and crossing it with the best and looking for transgressive segregates, things that would be even better. Okay? This is one of the challenges where something like yield is the major objective. You can take something that's high yielding and something that's high yielding, and you know you'll get something high yielding out of that, but what you actually need is something better yielding than anything you put in there. Okay? So you're always looking for transgressive segregates. Backcrossing, in case you have a need for a particular trait. In that case, it's a little simpler. Maybe you've already got something that you're pretty happy with the yield, but it really needs a bump in disease resistance. Okay? There you don't need to find transgressive segregates. You just need to generate new combinations of genes where you can have both the yield gene complement and the disease resistance gene in the same pedigree. Okay? So there you're going to cross an elite with something that's donating the trait it needs. Yeah.
So just to get it right, an elite is that an elite inbred line?
Yes, in this case I'm talking about elite inbred lines. And how do you define them? Because they give you good hybrids. Okay, so elite inbred lines are defined by their performance in hybrids, not generally by their own characteristics. Okay, was that? Yeah, good. So you're crossing things that you know. In this case, if you're looking for improved yield via a pedigree approach, you're crossing inbreds that you know will produce high-yielding hybrids, and then hoping you get something that happens to produce an even higher-yielding hybrid out of it. Okay? Backcrossing, you're generally trying to improve a defect in a parent, okay, with a specific gene or a few genes from another individual. More recently, doubled haploids has entered the world of maize breeding. It's been around as a technique for a long time. It's essentially a way to generate instant inbreds. Okay? If you take anthers, pollen cells, or some other haploid cell type, and you can manage to regenerate plants from it, that's a way to get a whole spectrum of instant inbreds. The technique is still a little cumbersome and costly. It's not like you can take 10,000 pollen grains and get 10,000 plants. There's a lot of loss along the way, so generating those can be a challenge. There are seed companies who are doing maybe 20%. These are the large seed companies, you know, the Pioneers and Monsantos of the world, who are generating maybe 10 or 20% of their inbreds that they evaluate each year through doubled haploids. The advantage is it's fast. Okay, you culture them, you get the plants, you've got your inbreds. That was it. As opposed to an inbreeding process where you need maybe 5 or 6 generations of self-pollination to have something that's fairly homozygous and uniform. So it's fast. The disadvantage perhaps is you have no opportunity for selection along the way. So anything you might have selected for, you've still got all that garbage in the mix, right? If you've got a lot of garbage inbreds, you know, a pedigree always gives you mostly junk and a little bit of good stuff. You still have all the junk in there to deal with at that point. Yeah?
I don't totally understand the use of the term transgressive segregates.
Ah, okay. So transgressive segregation is simply, can we see the blackboard? Do I have the lights? Yes, I have the lights. Okay, cool. So if this were a graph, this is a frequency distribution, okay? So frequency over here, and let's just say yield over here, all right? If you have progeny, like hybrid progeny from one parent that give you a range of distribution like that, and hybrid progeny from another parent that give you a range, there's the mean of distribution like that, you would expect if you cross those two parents, you would get some sort of a range that covers the two of them, but typically, you'll also get things that fall outside the range of the two parents, okay? These are the transgressive segregates, okay? So they're things which fall outside of the range of phenotypic values of the parents. usually, there's always ones down here, and usually a lot of them. Those are not the ones we're looking for. We're usually looking for the ones that are better, right? So transgressive segregation is almost always what your aim is when you're trying to improve yield, particularly. If that's the goal, you've got to always be looking for things which exceed any of the parents you put into the cross. If they don't, just go back and use the parents. You've already got them. They're better yielding, right? So if it doesn't exceed your parents, you haven't achieved anything in the world of yield improvement. So yield, we're always focused on transgressive segregates. Things like, adding disease resistance to a high-yielding progeny, that's just about getting new gene combinations and not necessarily about the transgressive segregation. Okay? Good. There we go. Typically, as you're doing this inbred development, before you get to homozygosity, you're going to have to focus on some traits that are high in heritability. Why? You're selecting single plants or single progeny rows in the field. If the trait isn't fairly high heritability, your selection is going to be meaningless. Okay? The low heritability traits, like yield, for example, all important, you can't really do much with either until the lines are fixed so that you can evaluate replications in multiple environments, and you can know that all of that dominance and epistatic variation is not segregating and recombining anymore. You now have fixed lines, so whatever they have is what they have. Or you can do it earlier in the process if you can figure out a way to replicate. Okay? So that's the challenge with inbred development. The most typical approach we use in the world of corn breeding is pedigree breeding, crossing that elite by elite lines and looking for things which are better. This is the general nature of a pedigree process. You create an F1, you self it to generate what I call S1 families. This would be the F2 generation or S1 plants. The F2 or the S1 is where you see everything that was heterozygous in that F1 segregating out. So this is your most variable generation. Every plant will be genetically distinct. The number of possible genetic combinations is usually astronomical. You couldn't possibly sample them all. So what you do is you sample, you generate S2 families from pretty much all of those S1s, because there's really nothing you can tell about a single plant that's all that useful. Maybe you can throw out a few that are really too late maturing or something. And then you start keeping track of sets of S3s. You're going to throw out some of the S2s that don't look good. You're going to look at sets of selves from each of those S2s in the next generation, the S3 generation. You know that these three are all pretty closely related, so you're not going to bother to keep all three of them, but you're going to pick probably the best one of each set. A few sets you might throw out. But the goal of this, picking the best one and then looking at another set of families at the S4 level, each level of inbreeding, those families are going to be getting more similar to each other because you have more homozygous loci in the predecessor, which are fixed, right? So they're going to start looking pretty similar, but you can always then pick both the best sets of families and the best family within that set. And ensure that at the end of the process, you've got a set of things. I've just stopped at the S5. That's a typical, maybe that's it. You might do a few more generations of inbreeding, but that's probably enough. At the end of the process, you've got a set of S5s, each of which traces back to a different original S1 individual. So you've sampled some of the variation in that population by ensuring that you're keeping a sample of a number of different individual parent plants. And then you can screen for a trait like yield, where what you're looking at in the parents themselves doesn't tell you hardly anything about the yield potential of the hybrid. If you can't tell anything about the yield potential of the hybrid, your best bet is to save a broad sample of the possible genotypes. And then you won't have lost by random chance the better fraction of the population. So that's what pedigree is about. It's about giving you an opportunity for some selection during the inbreeding process, and at the same time ensuring that you're saving a fairly broad sample of individuals. Contrast that, if you weren't doing this consciously, maybe the progenies of this S2 just happen to look really great. They look nicer than anything else in the field. So you keep those and you throw everything else out. What have you got then? You've got a whole bunch of stuff that is very closely related. If that genotype doesn't happen by chance to also have decent yield potential, you've lost the best possible segregates out of that cross by not maintaining a diverse range for the trait that you can't really see or evaluate until you get to homozygosity. Okay? So that's the trick with Pedigree. It gives you a way to ensure that you're maintaining variability for traits you can't yet evaluate, and at the same time giving you a chance to select for the traits you can during the process. Backcross breeding, again, to add one or a few traits to an elite inbred, it's something you can do fairly rapidly. You can run your generations quickly. You can do it outside of the area of adaptation because you're predictably coming back by backcrossing repeatedly to that elite inbred type. So there's no need to really select for all those background genes. The only thing you have to select for is the trait from the donor. You can use markers to do this more rapidly. If you genotype your recurrent parent with markers, you can actually use the markers to select for that recurrent parent genotype and get, with only 2 or 3 backcrosses, to the same place you would get with 5 or 6 in the absence of markers. Downside to backcross breeding is that your elite inbred that you used after a few years may be bypassed by new inbreds that are better. So by the time you get to the end of the process and you've added, say, a disease resistance gene into this good inbred, you might look at it in your hybrid combinations and say, oh yeah, but the inbred I just developed in my Pedigree program is even better yielding than that. So I wish I had my new disease gene in that one. So since you do all of these things kind of in concert, your pedigree improvement is continuing, your backcrossing will get you back to a fixed inbred type that is no better in yield potential. So if you've gained too much, if your pedigree breeding was too good, it'll leave the backcross product in the dust, okay? So this is why particularly in the private sector, companies try to do this very rapidly. So it only takes them a couple years. That way they haven't, their inbred recurrent parent has not been bypassed by their ongoing breeding effort. Interesting that about 90%, I believe right now, I should check that figure again, but maybe 90% of the marker use in commercial maize breeding is used to accelerate the backcrossing of transgenes into inbreds. So in the industry, a major priority, you do your breeding to develop a new hybrid, and then you want to add the Roundup Ready gene, the BT gene, the BT rootworm gene, this suite of transgenes. The most extensive use of molecular markers in the private sector, so in commercial breeding, is simply to rapidly backcross those transgenes into known inbreds. So marker-assisted selection in terms of improving traits has yet to really be implemented in corn private sector in a big way. They use a ton of markers. There's lots of money that goes into it, but it's mostly about moving transgenes around as quickly as possible. So just point of interest. So inbred development, I said you want to select initially for high heritability traits that will be useful. These are things that when you see them on the inbred plant are likely to be passed on to its hybrid progeny. What is high heritability? By definition, it means you have lots of additive gene action. What is additive gene action? I always like to define it as what you see is what you get. The parent, if it has early maturity, will pass on those additive genes to its hybrid progeny, and the progeny will also have early maturity. Okay? So things like the appropriate maturity, disease resistance, nice differences. That's a difference at flowering time for a disease called gray leaf spot. Anybody who's not even a maize breeder can probably tell which one to choose there. Stalk quality, it's important, right? It's something you can evaluate on inbreds. And also ear and pollen traits that you need for seed production. Those are about the only traits where the inbred you're looking at is an accurate representation of the product you want. You want an inbred that you can produce seed on to market to growers, okay? So those are important traits. You want to evaluate, despite this, you do want to evaluate yield potential early in the process because it's the most important trait. How? A couple ways. You want to know your tester in advance. And what I mean by that is when I put together a cross to try and get a better yielding hybrid, I already have in mind what genetic family it will combine well with. So we know a fair amount about what families of things make good hybrids in the world of corn. I already have a tester in mind, in other words, the other side of a hybrid in mind when I make a cross. This is because we know a fair amount about maize heterotic groups. Heterosis is hybrid vigor. Heterotic groups are just groups that have different genetic origins that tend to give you high hybrid vigor. If you don't know anything about a collection of germplasm, maybe you're working in a new environment with a lot of farmer varieties, simply picking things that have different ear shapes, long thin by short fat, is a pretty good way to guess which things might give you heterosis. Why? Well, maybe you'll get the short thin ears, but you hope you'll get the long fat ones, right? So that might give you good hybrid vigor. Pedigree relationships is the other way we know about this, and this is certainly the case in the US today. We have a lot of knowledge of what genetic types give us good hybrids. We know which inbreds are derived from them. We have built those heterotic groups through breeding. By selecting constantly for things which give you higher and higher yield, you have built blocks of genes on both sides of that pedigree that are complementary. Okay? In my breeding program, I basically work with 3 different groups. You don't need to know the names, it's not important, but there's a group of materials derived from things related to the stiff stock synthetic, another group with iodine, and another group with W153R. I know that combinations between any pair of these tends to make higher yielding hybrids. So when I make a breeding cross, I'll either stick with crossing a couple parents that are both stiff stock and aim to cross that to one of these 2 testers, that will be the other side of its hybrid parentage. Or I might actually cross some of these together, but know that this is going to be what it might make a good hybrid with. So I already know that before I even start. Okay? Then what I can do is when I've gotten either the F1 or the F2, one approach is to take that and cross it to that tester and see whether it has any yield potential to begin with. If you haven't got any good yield potential in the mix, then selfing out of the F1 and F2 is not going to give you genes that weren't already there. You're not going to recover it if you don't have it. Similarly, but a slightly different approach, you could use early generation tests, and that's what this looks like. You start your pedigree process, but maybe when you get to the S2 level, you cross each S2 plant to a tester at the same time as you self it. And then you do a yield trial with the test crosses, and you continue selfing with the selfs. You can do that at each step of the way, which is not unusual in the industry. So you're selecting things which you know at the S2 level have some genes for combining ability in hybrids. You don't have to keep selfing them if they don't. And then you're going to do the yield trials later on in the process. I see I'm running out of time, so what I'm going to do is just talk about the next 2 slides, and then we'll wrap it up, and that will at least have given you the guts of corn breeding. So inbred development, you want to select high heritability traits that will be useful. You want to evaluate yield potential early in the process, given that it is the most important trait we're dealing with. And what yield evaluation of experimental hybrids at the end of the process looks like is sort of like a funnel. You start out with a lot of hybrids, and you put them at relatively few sites and reps because you can't afford to put them at more. At the bottom of the funnel, you have very few hybrids, but you're evaluating them at lots of sites. So the actual number of plots you're putting out might be pretty constant, but the number of genetic materials is funneling down very rapidly. Just to show you what this might look like for an actual example from one of the large private sector breeding companies, this is a company that just for the US market tests about 50,000 to 60,000 new hybrids every year. So these are finished inbreds, they've made new hybrid combinations, and they're going to put in yield trials 50,000 to 60,000 new hybrids. Admittedly, they have a number of breeding. This is not one breeder. This is like 10 or 15 different breeding programs at different locations, collectively among them. By the 3rd year of testing, they're only going to have about 2,500 of those still in the running. So that's about 5%. So you've thrown things out very rapidly, okay? And ultimately, fewer than 10 might ultimately get released. So you're sampling an enormous range of these inbreds for hybrid yield potential, the all-important trait, but the one that you don't know much of anything about until you actually test them. And you're throwing things out very quickly. I want to just end with a nice quote from Allard about this process. This is the old Allard, but he wrote very nicely. The beginning plant breeder must develop an attitude of ruthlessness in selection. He must dispel the feeling that among the plants he discards may be the one that will lead to the variety he has in mind. Unless he keeps this natural inclination under control, this is all he's noticed, he will shortly find himself overwhelmed with material that he should have discarded, and his effectiveness as a plant breeder may be impaired or lost. So just remember, in selection, the game is all about throwing out enough stuff And unless you're kind of ruthless about it and lose the idea that, you know, that one plant out there is, that you haven't found is the one that's going to save the world, you'll be buried in stuff. And that's an example of how fast you might throw things out. Thanks.